ichthyology or fish collection curator. So, all right, so this whole call and funding was all about microplastics, and seeing as I'm the first person up, I figured I would take a second to just say what microplastics are for everyone on this call. And that is any plastic particle 5 millimeters or smaller as defined by NOAA. There is a lot we don't know about microplastics in wild environments and how animals, including ourselves, interact with and deal with microplastics. This is from the Track 1 funding call, which is to improve our understanding of aquatic microplastic sources, and uh, particularly for this project, the ecological sensitivity of microplastics. In terms of ecological sensitivity, there's also a ton we still don't know about microplastics and wildlife. We don't know when animals first started getting um, affected by microplastics, either consuming them or co-occurring with them in the environment. We don't know if microplastic consumption is increasing over time. Has it been relatively steady over the past few decades? It's unclear. And we don't know even some of the most basic things. Are there differences between species in how they interact with microplastics in the wild? If there are, is it because they have different feeding ecologies? Or is it because they live in different habitats? All of these are still open questions, largely. And so conceptually, what we wanted to do is fairly simple. We wanted to look at how microplastic consumption varies across a variety of different fishes along the California coast. So we have three main questions that we're interested in, and that is, how does microplastic consumption vary by species? We're going to look at several different species. Which types of microplastics are consumed? This helps get at the part of the Track 1 funding call that looks for aquatic microplastic sources. And we are also very interested in how microplastic consumption has changed over time and how this relates to each species ecology, how they feed, which habitats they live in. It's hard, though, to go back in time. We don't have an active working time machine where any of us can jump in and go back and take samples to look at. However, we can get around this because in my world, working at a natural history museums are the active or only active working time machines on our planet. So within any large major natural history museum with a research focus, there are a variety of collections that are full of specimens. These specimens document life on our planet, both past and present, and really show us how life has changed, both on the very big picture grand scheme of things. Think of seeing dinosaurs and how they've evolved, or how life has changed from dinosaurs to present. But even on small time scales, natural history museums work as a time machine as well and can show how humans have impacted wildlife on our planet. The Natural History Museum in Los Angeles County, where I work, is the largest natural history museum west of the Rockies. We have over 36 million artifacts and specimens within our collections. This project specifically focuses on fishes in our fish collection. This is a picture of our fish collection. It is in one room, and it's 3 million fish specimens spread out around 175 or 176,000 jars. Each one of these jars contains fishes from a very specific location and time. Each one of these has great records, so we know who collected it, when it was collected, what depth they were collected at, where it was collected. All of that information is stored on all of these specimens. So this is where we use, or these are the specimens that we use to actually go back in time and see if microplastics are in these fishes from different time periods. However, even though we have 3 million specimens in our collection, we cannot do this alone. And I think this really highlights the importance of natural history museums writ large across our country and the world. And this project has heavily been influenced and helped by the Scripps Institution of Oceanography, their marine vertebrate collection, and also the Ventuna Research Group, at Occidental College. We've all collaborated to provide specimens for this project. So we wanted to look at a variety of different species that occur in different habitats and that have different feeding ecologies. So we've actually targeted six in two different habitats. So one habitat that we're looking at is shallow water coastal habitat. And these are our four species that we're looking at in that habitat. We have shiner perch, northern anchovy, the opali, and the California halibut. These are important ecologically and also for some of these species for sports fishing as well. Each one of these fishes eats different things. Some are more generalist or invertivores, some are planktivores. We have an herbivore, the opali, and also a piscivore. We can contrast these species too to also a different habitat, our deep water habitat off of California, where we have two mesophotic species, the northern lampfish and the Pacific black dragon. These are mesophotic planktivores and mesophotic piscivores, respectively. So we can at least compare these two feeding ecologies to shallow water planktivores and piscivores to see if there's any difference between habitat deep or shallow. This is where we've taken samples from. And it looks a bit messy, but I wanted to show this to highlight that uh, all of these museum specimens, they weren't collected for the purpose of this project. So we have to deal with the samples that we have. So there will be some spread. For all our shallow water habitat species, though, we kept them to coastal California and avoided any of the offshore islands. The deep water fishes, of course, do have to come from a little bit further offshore. So we have samples from both the San Pedro Basin and the Catalina Basin as well. For each fish, we dissect out the guts, we digest it, filter that, we image it using optical microscopy, and then we use Raman spectrophotometry to actually identify what types of plastics are in the sample. We do a very fine search on our filters, searching every 15 microns essentially, to try to find as many small pieces of microplastics as possible, because microplastics can keep on breaking down from that 5 millimeter threshold. They can break down further and further and further until they even become nanoplastics. But nanoplastics are out of the range that we can detect with our methods. All right, so some brief results. Again, this is preliminary data, as has been said. These are not peer reviewed. And in fact, this project is still ongoing. We got an extension on our project, and we will be continuing to process data over the next year. However, there are some things that I feel confident showing and sharing with you all today that I don't think are dramatically going to change with more data coming in this next year. So we see plastics as early as 1949 in our samples. These early plastics are mainly acrylic and polyethylene, which were both invented in the 1930s. Both were used heavily in the war effort in the early 1940s and mid 1940s, and then became commercially available after that as well. So we start to see the influence of that pretty quickly in our specimens, starting in 1949 that we see plastics. We find plastics fairly commonly by the late 1950s and early 1960s, and every single species that we've looked at has microplastics. There is no species that avoids consuming microplastics. These are some very common plastic types that we see pop up time and time again, especially polyethylene. Polyethylene we see a ton of. It is in many, many samples, not every single sample, but it is in the majority of samples. Polyurethane, polypropylene are also very common. And we also have, the fibers that you're going to hear about later in some of these other talks, such as nylon and polyester, those pop up as well. Interestingly, though, there are some things that did surprise us. Plastics are not in every fish, even from really recent timescales. So even, last decade, we have some fishes that are actually empty of plastics. But some fishes are chock full of plastics. So the image that you can see on the screen is a map from our Raman spectrometer. And this map, the colors, the red and purple that you see, those are plastic particles. This is a fish from the 1960s, and it is just full of plastics. Obviously, if a fish's gut is full of plastics, that can have adverse effects with how that fish can digest food. It can lead to nutrient deficiencies or even death. But at least on the somewhat positive side of things, most fishes aren't jam-packed full like this one sample. Plastic loads, although common in many individuals, are fairly low. Uh, and again, some individuals don't have any plastics in them. Uh, this is even true with sometimes we'll sample multiple fishes from the same jar, meaning they were collected at the same time and place. There are fishes that were schooling together. And sometimes you have fishes from the same time and place where one individual will have plastics in it and another individual will be completely clean. So this could imply a couple of different things. Either fishes can behaviorally adapt and some fishes don't eat microplastics, or that these fishes can actually clear their guts. And again, this project only is looking at the digestive tract of these fishes. It is not looking at accumulation in other tissues, which could say a completely different story. Um, Again, this is preliminary data. We do see on average, though, an increase in microplastics across time, although not perhaps as strongly as you might anticipate. But again, what I'm showing here is very preliminary type of results. This is actually not how we're going to analyze the data in the end. This is just for illustrative purposes of a single species. But again, we're going to be comparing many different species. However, we don't have the data to where I am confident that I could show you results that don't dramatically change in the future. So this is, but I will say that we do see a general increase over time in microplastics, which is probably what you might expect. A major part of this project, though, has also been outreach, and I will go through this relatively quickly because I'm running out of time. But we have done a lot of outreach at the Natural History Museum, and we will continue to do so over the next year. We've had a partnership with Nickelodeon over the past, and we have actually a YouTube video up where we talk to a young Nickelodeon host about microplastics. We do Earth Day every single year. This is a single day where about 3,000 visitors visit the museum of all ages, and we have a booth specifically on microplastics. This next upcoming March, we will do a school program at the Natural History Museum called Collections Revealed that will be all about microplastics. This will target school-age children from LAUSD and other schools in the area, and we expect to see around 800 people total over the month of March that will be exposed to this. And we will also have a temporary exhibit up at the museum later on this year. This is not that exhibit. This is the finalization of a previous temporary exhibit, but gives you the idea of kind of the size and scope of an exhibit that we will have. So the Natural History Museum on a good year sees up to 1 million visitors annually. And so a temporary exhibit will be able to display our results to the public and inform the public to at least several thousand visitors during that time. So why does this project matter? We still have a very limited understanding of how wildlife interacts with microplastics. Very few studies have been done. Many are limited by the species that they look at and location. We still don't really know general trends of how a species ecology might impact microplastic contamination. And this project is particularly important because it's hard to judge change and also mitigation efforts, if they're successful or not, without having any baseline data. And this is the only way that we can go back and really see how microplastic contamination in our wildlife has changed over time. All right, so I'll be happy to take any questions if there's some time.
Thank you so much, Bill. Yes. Um, we don't have any questions in the Q&A just yet, so if anyone has questions, please enter them in the Q&A function. You might need to press on the button that says more with a few dots to open that feature. But we do have a question from a fellow researcher. So Matthew Savoca asked, are these fish microplastic data all gut samples? Or are there any samples from other tissues of the fish?
Yeah, so that's a great question. It's something I think about a lot. These are all guts, just digestive tract. That's what the funding for this was for. Um, I would be super interested to look at other tissue types within these fishes, and especially because we're using catalog museum specimens, we can in the future go back to the same exact specimens that we have gut data for and look at brain or liver or muscle. And so you could see a really interesting pattern. Does the gut data reflect other tissues that might accumulate and not shed plastics? Or there's a lot of open questions that this research has already gotten me really excited about.
Very cool. I love this project, Bill. We'll be in touch at some point. It's really cool stuff. Love the museum aspect. Great work.
Love to hear it.
Thank you, Bill. And unfortunately, we are running a little behind time, so I might ask there are some Q&A questions coming in the Q&A if you might don't mind just taking a look at that and answering them. Thank you.
Will do.
All right. Our next presenter will be Matthew Savoca from the California Marine Sanctuary Foundation, sharing his project titled From Watershed to Whales, Tracking the Source and Transport of Microplastics in the Greater Monterey Bay region to inform
Can
risk assessments.
can we see that? Can everyone see that?
Yes.
Okay, cool. Thank you very much. Thank you to the funders, to those here today, and of course to our collaborators on this project. This has been a fun effort. As Bill mentioned, it's still an ongoing effort. Almost all the samples are collected at this point, but we are working through the data. We have a lot of data already analyzed, and but more coming in, and we also have a 1 year extension to deal with all that data. But this was a really exciting project and by far the most comprehensive work done in the Central California region, understanding where microplastics come from in our ecosystem, where they go to, and the risk they might pose at the land-sea interface. And so this picture here was taken by one of our participants or one of our collaborators, Chad King at the Monterey Bay National Marine Sanctuary. And I really like this image for this project because it shows actually kind of several of our sites simultaneously. It shows the San Lorenzo River moving moving right to left, the San Lorenzo River, the Santa Cruz Boardwalk, the Santa Cruz Beach there, and the ocean out into the Monterey Bay. And so that's sort of the ecosystem that we were trying to capture, and I think this picture does it pretty well. So I work for the California Marine Sanctuary Foundation, the lead organization on this project, but we have other incredibly important organizations that were part from the beginning, the Monterey Bay National Marine Sanctuary and the Moore Institute for Plastic Pollution Research in Long Beach. We've since worked a lot with CSUMB, that's California State University, Monterey Bay, and I also work at the Hopkins Marine Station of Stanford University. So one of the great things about this work, I think, is the community that it has afforded us to bring together on this effort. And I'll explain to you some of that work that we were able to accomplish. So first of all, just to get started real quickly, because I know these are short presentations, what our project objectives were. First, to understand the transport and basically the flow of microplastics between rivers, so freshwater, terrestrial environment, out to the beach, the ocean, and into the wildlife in the Monterey Bay. We want to try to do what we can with the data at hand to understand sources of microplastics to our nearshore ocean, which is much easier said than done. We'll probably have a discussion about that later on, I would think. Three, we wanted to engage a bunch of citizen scientists in this work, and we were able to do that. I think over 100 citizen scientists have participated in this field work, and I was on a local, just randomly on like a local Nextdoor group or something, and I saw people talking about it, like, oh, this is a really cool environmental project you should volunteer with. So I'm like independently seeing it come up in some of our local environmental circles, which is pretty cool. And then finally, this is thinking about this ecosystem as sort of a test ground or test case for how we can understand plastic pollution in ocean environments, and specifically where plastics end up in ocean environments and the risks they might pose. And the Monterey Bay, we think, is a great exemplar to sort of study those processes. The project consisted of a lot of field work, which actually started even before some of the funding came in, because we were excited about it. So we started, testing ideas to sample some of this microplastic. One of the cooler ideas that came from this is Pam Crone. You could see up in the top left-hand portion of your screen there with Wynn Calgar and Chad King up on the bridge there, and sampling microplastics from 50 or so feet above the middle of a river. But came up with innovative solutions to sample rivers here. That's a peristaltic pump that we use to sample all the rivers in our ecosystem. So that pumps hundreds of liters of water an hour through a stacked set of sieves, and the stacked sieves is what we actually analyze for plastics. So then we also measured a ton of beach sand. As far as I know, there's been very little, and actually, as far as I know, no studies on looking at microplastics in beach sand in this region. So this was the first study, as far as I'm aware, to do that, and we found a lot of it and tested a lot of sand. The picture in the bottom middle is from seawater sampling off of the Santa Cruz boardwalk, as you can see in the background. And then in the top right, that's kind of my bread and butter. That's our lab mate Jack Barkowski at Stanford collecting a nice blue whale poop. That's one of the best examples of blue whale poop I've ever seen. So I was really excited when he found that sample for us. And so, in some of our samples, this is sort of a range of what some of these plastic particles look like, the ones that we can see anyway. These are some of the larger microplastics that we see. So, in the lab, we separate using those stacked sieves samples into larger microplastics, which is to say 5 millimeters to 500 microns or half a millimeter. And that's the particles that you see in the bottom right here. Those are ones that you can actually see with your naked eye. And then smaller than that, we are testing down to samples that go down to about 50 microns or maybe even a little smaller. And those are samples that I can't show you exact pictures of like this. I'll show you images from the micro FTIR from, but you can't see those really with the naked eye. So, but this is kind of an idea of what some of those little microparticles look like. Most of those samples in the bottom right there are from a beach sample in Santa Cruz. So just to get into the data real quick, I realize that this is a short presentation, and so there is a mountain of data that we're sifting through now, and I'm kind of just showing you a bunch of summary slides of those data. There's a lot of inferences that we're starting to draw from that information, but at this time, and particularly because of the brevity of this talk, I'm just going to kind of go over it at a high level, but of course, happy to answer questions you all might have. So a general theme, which you'll see throughout the results, is that beach sand, and this is one of our higher level results answering one of our objectives, beach sand seems to be both either a source from the people that use the beach and certainly a sink of microplastics in our ecosystem. So these data from this first slide are just from that larger size class of plastics. These are the plastics in the millimeter size range, things that you can see with your eye. And again, you see when you look at the rightmost column there that microplastics in beach sand are about 20 times higher than in beach sand than they are in ocean water, river water, and so on. Also, importantly, our blanks are pretty low, so that's good generally. So just some summary data of this larger size class of plastics. Some interesting things. Most of this lines with what we've seen in previous studies from around the world. Almost all of the plastics that we find are either white, blue, or black. So that's kind of interesting, even though there's lots more colors in the world. Almost all of what we see in the environment is white, blue, or black. We see a lot of fibers, and fibers tend to be smaller than fragments. So most of what we see are fibers, films, and fragments in terms of microplastic morphology. And you get a sort of a size distribution of those different particle types. And then looking at these plots down in the bottom, if you go from left to right, so left just looks at basically an absolute count of all the particles that we found in the larger size class for these different samples so far. What you'll notice is that there's a lot more plastic in beach sand. Not only that, but it's basically the only place we find foam. So expanded polystyrene of some sort. The only place we find it, and we do find it quite a bit, is in beach sand. So I think that's an interesting potential route for mitigation, which we can talk about later. And then moving over, for example, and then the middle plot is just those data transformed to percentages to easily see them. And then moving over to the bottom right, what you see is two of our watersheds are agricultural in nature. So that's the Salinas and Pajaro River watersheds. And two of the watersheds are urban in nature. That's the San Lorenzo and the Carmel River watershed. They're not dense urban centers, but they're more urban identified watersheds than agricultural. And so you can see that basically urban watersheds seem to have more plastics than agricultural watersheds, at least for this larger size class. Moving to the smaller size class, these are particles that are, in general, the width of a human hair or smaller. Human hair is about 100 or 200 microns or so. So that's just giving you an idea of how small these particles are. And in this size class, this is where most of the action is happening. And I would imagine that if we were able to get more funding to do work looking at even smaller particles, these numbers would be even larger. That's generally the trends that we've seen. And so here again, you see though the general pattern that beach sand is much higher than river water or ocean water, and also higher than our blanks, luckily. This is an example of what sort of a mapped a particle map from one of our filters looks like from the micro FTIR. So this is just one filter from one sample, and there's over 600 particles on that filter. And the FTIR does really cool work because it doesn't just identify plastics, but it also identifies organic matter, minerals, things like sandy bits, stuff like that. And so doing that, you can actually look at the entire spectrum of what's going on in the particle space, the small particle space, in these environments, in these ecosystems. And so these are all of these samples that we've processed so far. Again, we still have some more to go, but I'd say it's about 80% of our final data here. And you'll notice a couple things. One thing that you might notice is that while there are a lot of plastics in these samples, they're not often the dominant particle type in our samples, right? So we see more organic matter and minerals than we do microplastics in most samples, so that's a good thing. However, there are microplastics in basically every sample we look at, ranging from just a few particles to a few thousand particles. So, this is of course, we want to know within that range, where is the concern lie? Where does the concern lie? So with the micro FTIR and those 23,000 particles that I showed you earlier, it identifies more specifically what polymers those particles are. And so even though we've identified over 24 different types of polymers in our environmental sample so far, almost all of them are these 5 polymer types: polypropylene, polyethylene, polystyrene, polyacrylamide, and polyester. Again, this is not terribly surprising, actually. People who work in the microplastic space would know this, and if you don't, then this is something that we see commonly in microplastic land, even though there are hundreds of different types of synthetic polymers in our world. Most of them are from these dominant polymer types. And so, but there are some interesting things going on here. So, for example, what we find most commonly, or what we've found most commonly so far, and again, these data are preliminary, just like Bill said for his project, so I wouldn't, run and tell the press about this just yet until we are able to publish it, get it peer reviewed, and so on. But in any case, we see polypropylene so far as by far the most common polymer type in our environment. This is corrected for blanks as well. However, if you look at the global production of plastic, only about 18% of plastic production is polypropylene. So that's really interesting. That implies that something is happening between the production of plastic and the environment, at least in this ecosystem, of why polypropylene is more prevalent as waste than it is in its relative to how it's generated. And this is also generally a trend that we see globally. So waste generation by polymer in 2019, polypropylene again is the highest of those polymer types that turns into waste, even though it is less than a fifth of global plastic production. That being said, it is the most common of any polymer produced, but not by the margin that we see in environmental waste all the way down to the microplastic level in our own ecosystem. So these are some interesting sort of early things to think about, big broad picture stuff. So thinking about risk assessments, we can start to put our numbers into frameworks, updating prior numbers from our ecosystem. So we're finding higher levels than in the past, and there's lots of reasons for that. Obviously, a lot of time has passed, certainly since those first samples, but also we're sampling for smaller particles, and the smaller particles there's lots more of, as we talked about. And so thinking about how these levels plug into toxicity thresholds will be interesting. So these are some proposed toxicity thresholds from Squirp and Alvamanhito et al. In 2022. And so the levels that we're finding in seawater, again, preliminary, ends up being at this level of potential food dilution at these lower levels of monitoring to instigate. We should basically be monitoring this problem, which I would agree on. However, as I mentioned, for the beach sand stuff, and I don't know, this would be something that I want to talk with some of the management and regulator type folks on the call today. For things like beach sand, we see levels that are way higher. Now, of course, these levels here for food dilution and tissue translocation, those are water-based thresholds, I believe. So to how they might translate in terms of risk for organisms living in the sand or feeding in the sand like shorebirds do, I don't know, but we do see much higher levels down to that, around 30 particles per liter in beach sand. So you may have noticed that our project is called Watershed to Whales, and I haven't yet mentioned the whales or the wildlife that we're sampling. That's because those samples are really, really hard to process. They're hard to get, but they're really fun to get, as Jack shows you here. But they're very hard to process. So we're in the process of dealing with those samples now. We are finding plastics in them. This is a pretty nice microfiber that we found in a blue whale sample, blue whale poop sample. That's what it looks like under a microscope, if you were curious. But we haven't yet sort of processed those data to the point where I'm confident showing you numbers yet. But we are finding them in most of our samples. And what we find is sort of generally the same as what other studies have found around the world. Our numbers might be a little bit higher, again, because we might be looking for smaller particles or things like that. But in any case, sort of generally works with what has been found around the world. So to wrap up quickly here, because I know that we're short on time, I just wanted to go over some key takeaways of our work so far. One is that we find far more particles in our lower size class than we do in our larger size class, I imagine. And it's not linear. It's sort of more tending more toward exponential. So as you get smaller and smaller, we're finding potentially exponentially more particles. And I think that trend would probably continue if we went to even smaller particles down to the single micron or even submicron size. But that has to be for future work if we go there. Understanding flows. So basically, again, beaches seem to be a major sort of sink and source potentially of microplastics in this environment. Concentrations 20 to 30, sometimes more, 30 times higher than our other abiotic components of the environment. And microplastic levels in all of these abiotic components are at or above the lowest risk thresholds for things like food dilution and organisms living in those spaces. Even though we have found over 24 different types of synthetic polymers identified by our micro-FTIR, almost all of them are just from these 5 polymer types, as I mentioned earlier. And an interesting finding is that we find a lot of expanded polystyrene, which is not technically styrofoam, but like colloquially we would call styrofoam, on our beaches. And it's found almost nowhere else. So this is kind of interesting because, for example, in Santa Cruz, there's been an ordinance preventing, for example, single-use takeaway styrofoam containers for over a decade now, and yet we still find it on the beaches. We don't know exactly where it's coming from, but it's still there in some abundance on our beaches, particularly in the Santa Cruz area. And we're working on the biota data, as I mentioned. We're starting now with a paper focused on the transport of microplastics in our rivers. And we've already submitted for some follow-up funding to sort of do some sourcing. I tried to work on some sourcing and chemical risk assessment of the particles that we found in this project. So this project is already feeding in, hopefully, to future work that we can get funded to move on and build off of it. And Bill mentioned at the end of his talk how there was such engagement with this issue, and we found the same thing, right? So we have people that stop us as we're doing the work on the side of the road or on a beach and saying, "What are you doing?" And then when we tell them, they're so thankful. And not to mention, we have again over 100 citizen scientist volunteers, and so. With that, I just want to go very, very quickly into acknowledgments. There's all the folks that we work with or have let us work in their spaces, like my Stanford supervisor, Jeremy Goldbogen, in the top left there. We have the students that I've worked with that have been absolutely incredible. Corey Lopez presenting some of their work at a CSUMB research showcase just below. Getting to work with Wynn Calgar and the Moore Institute folks in Long Beach in the bottom left has been a phenomenal experience. I've learned so much from them. And yeah, it's just been a really wonderful time, and the people are a huge part of it. So maybe the biggest part of it. So that's been quite a joy, and I want to thank the funders for giving us that opportunity. And so yeah, so with that, I'm sorry if I went over, but that's what I got. And I don't know if we have time for questions. I probably went too long, so I'm sorry, but I tried to make it short.
Thank you, Matt. I know you have a lot to share in such a short amount of time, so we really appreciate you being succinct. I thought you gave a great presentation. We'll have one question. We did have someone come through, I think more of a clarifying question for you. Someone asked, when you mentioned that microplastics increase exponentially as size decreases, do you mean that the number of particles increases or the surface area increases? Can you maybe clarify that?
Yeah, good question. We're talking about number of particles, but surface area would be something that we can and would also look into as well. It's a good question.
Great. Well, thank you so much. We do need to move on. If any other questions get added into the chat, would you mind just responding to them? Thank you so much. All right. We're going to move on to our next presentation. It's the final presentation for this track. Lastly, but not least, we have Diana Lin from the San Francisco Estuary Institute representing her team and their findings from their project titled Investigation to Assess Clothes Dryers as a Source of Microplastic Pollution and Inform Solutions. All right, take it away.
Are you guys seeing my slides?
Yes.
Great. So thank you all for being here, and thank you OPC and Sea Grant for funding our study and giving us the opportunity to do this study that we've been wanting to do for a while, and thank you all for tuning in. So I'm so glad Matthew gave his presentation before because he pointed out that they saw a lot of fibers. And that it was a major motivation because we also have been seeing a lot of fibers in our San Francisco Bay samples. And one of our hypotheses is, well, one of our major questions is where are all these fibers coming from? Um, and so that's the motivation for our study. Are dryers a major source of microplastics? Um, as I said, this question has been brewing for a while as we've been studying microplastics in the San Francisco Bay. Our previous study, we compared microplastics going into the bay from wastewater and urban stormwater runoff. And we saw much higher concentrations of microplastics in stormwater runoff. So stormwater runoff is when rain falls on our urban landscape, it picks up contaminants, including microplastics that are on the landscape and transports them through storm drains and various flows to receiving waters like San Francisco Bay and other oceans. And so as we were thinking more about the stormwater runoff samples, about half of the microplastics we were seeing were fibers. And so we're like, where could these be coming from? And as we're doing our literature review and trying to develop our conceptual model as to what are the major sources, we came upon this hypothesis, like, is it dryers? Because it's not laundry, because that's goes down our wastewater and that's a separate sewer system than urban stormwater runoff. And so we were really excited about this funding opportunity and assembling our dream team to do this study. So we partnered with Monica Arienzo from Desert Research Institute and Lisa Ertl from Five Gyres, who have also been contemplating this question and doing related studies. And so for this study, our study objectives were to collect and quantify microplastic emissions from dryers. So this is the stuff that escapes the lint capture that is in your dryer, but like gets vented outside. And so once it's outside, there's no treatment for it. And we wanted to be sampling real life conditions so that we can estimate kind of what a realistic loadings is to the environment. And we wanted to answer the question, is this amount big or small? Like, is it significant? And we also wanted to evaluate some potential solutions because if we identify this as a major source, then we can start strategizing what are ways to mitigate this. And so Five Gyres led the effort to evaluate these secondary retrofit devices that can capture fibers before they get emitted into the environment. And our overall goal is to provide actionable data for managers. And then this picture is my colleague Kaylee Patterson's drawing rendition of our study design where there's the microfiber fuzzball trying to escape and we're trying to capture them. So this is an overview of our study design. We recruited community scientists to participate in our study, and we asked them to do their laundry the way they would normally do and install these. This is like a pest trap, and then we sewed a mesh that they would install on the device, and then do a normal laundry load. Tell us about what you're laundering, what you're drying, how much it weighed, and so we got a total of 55 of these samples. But not everyone has their own laundry machine in their house. And so we also sampled at a community dormitory laundry center at a local university. Different household sizes will do different levels of laundry. Like the more people you have, you typically do more laundry. And so we developed a survey that we shared widely to find out kind of on average, how many laundry loads do people typically do? How many people are in your household and try to do a correlation with that. And our samples were analyzed by Monica Arienzo's team at Desert Research Institute. And these were very carefully and tediously analyzed for microplastics for mass count and identified with FTIR spectroscopy. So what was the stuff that we saw? Well, interestingly, most of the microfibers were cotton, and a much smaller percentage were plastic, kind of like Matthew was pointing out how a lot of the stuff that they're seeing were also not plastic. But to me, this is really interesting because it tells me that cotton is part of the microfiber emissions, whether you class it as plastics or not. And then to kind of extrapolate these results to find out whether these are big or small, we took these results, we calculated average household emissions. The average household did about 230 dryer loads per year. We extrapolated this to the Bay Area watershed population, about 7 million people, 3 million households. And so this would be the emissions of like microfibers onto the landscape. And then a fraction of that will be picked up by stormwater runoff when it rains and get transported to the bay. So what did we find? Like, was this big or small? So these are preliminary results, but to give you a sneak peek, average emissions about the amount of microfibers that are emitted per laundry load was about the mass of a pill. If we, for the whole year, plastic emissions about the amount of like a heavy piece of paper. And then if we extrapolate this to like all of the plastic microfiber emissions onto the watershed in a single year, about the mass of an African elephant. And then a fraction of that is transported via stormwater runoff, and is about a moose load amount. So very significant. And so now that we know that this is a major source, what are potential strategies? Well, like we could brainstorm, are there ways that we can design our clothing to shed less? Can we not vent our dryers outside, use different types of dryers that don't emit outside? Or are there retrofit devices that we could install on to dryers to capture the fibers before they get emitted? And so 5 dryers led the study, and they tested 3 different inline retrofit devices. And these are results. The purple is the control, the amount of emissions from the control when you don't install the retrofit devices. And then the other 3 boxes are after installing. And so you can see that there was significant reductions. And these results are published in a peer-reviewed publication. So those are hot off the press. We're really, of course, this is not great results, but it's good that we were able to identify a major source in identifying outdoor dryer emissions as a significant source of microplastics. And then now that we know, we can identify strategies to reduce these emissions. So again, thank you all for this opportunity to share these results. This study really is in line with our mission, the San Francisco Estuary Institute. We're a nonprofit research institute, and our whole mission is to implement science that can inform management actions. And with that, I don't know if we have time for questions, and this is a picture of the abundant microfibers that we've seen in microplastic samples.
Excellent. Thank you so much, Diana. Great presentation. We do have a question for you. Elizabeth has asked, she has a question about the cotton. Her understanding of cotton is that it is natural and will biodegrade. Your statement seemed to imply that you considered them plastics. Is this because they are seeing plastics entangled in natural fibers, or is there another reason?
Yeah, so my understanding of California State Water Board's definition of drinking water is pretty expansive. It can include natural polymers depending on how much additives are in it. And I think, I don't remember, don't quote me on the threshold, someone probably knows. Our textile fibers can have significant additives. I don't know if it meets that threshold or not. And so, yeah, I think it's kind of that's a really good question, and whether it should be included in microplastics, I think is subject to interpretation.
Thank you. One more question we have, and then we will move on. Peter has said that they have a condensation dryer, a Bosch. So it doesn't have a vent. It has 2 layers of lint filters. It seems that is better from the microplastics perspective. Do you think?
That's what I am thinking as well, because it's not vented outdoors. I think it's actually gentler on our clothing, so maybe it doesn't shed less, but I haven't done this study. And totally, it doesn't get vented directly outdoors. It goes down sewer to wastewater treatment plants that capture where that is treated, and significant amount of microplastics are removed.
Great. Thank you so much, Diana. We're going to move on to our final two presentations. Right. Yes. And so we're moving on to our second track. This was to evaluate microplastic removal efficacy of low impact development and structural best management practices. And so first up for this track, we have Sanjay K. Mohanty from the University of California, Los Angeles, sharing the findings of his project titled Design Factors Affecting Microplastic Retention, Removal, and Generation in Structural Best Management. Take it away.
Thank you, Tanya.
Yeah.
I'm grateful to have this funding because we work on certain ideas of microplastic transport that we didn't learn before. And I have been working on microplastic for the last probably 7 to 8 years, and these projects was really helpful in understanding how we can best design these stormwater BMP so that we can remove more microplastics. So this again, as you see that the goal of a stormwater BMP is to capture some of the stormwater and remove a lot of pollutants. Plastic happens to be one of those class of pollutants, which are particulates. So they are really well designed to remove most particulates. So the goal of this project is to understand, can they remove them? And there's a distinction between plastics and normal particles, that plastics are lighter, it has different shape and size, and it can be fiber shape. So we wanted to understand how that influenced their removal. So there are three factors or three objectives when we started this project. First, what kind of plastic we expect to find in the stormwater, and what are those, how they are being retained in the system. So land use is a big factor. If you design this BMP in some locations, you want to mention what kind of water, where it's coming from, so that you can design the BMP accordingly. And then we also wanted to understand if the BMP located in any specific location received disproportionately more plastic. So that's why we do monitoring on plastics at different BMPs. And then the very end part is mostly lab design. We didn't do any field studies, but mostly to see how we can design them such a way that you can remove more microplastics. So again, the first two tags is all about land use. We went to different locations in LA and collected dust, roadside dust, to see how much plastics are there, because these are the source. Then we went to multiple BMPs, which is basically already designed or being constructed. So we measure some of the microplastic in those locations. Then we did most of the lab studies, designing those studies. So first results, how the land use affects microplastic concentration in Los Angeles. So here we went to many sites in our roadside environments, particularly the sidewalk, and we collected this dust. And we initially thought that the dust, the concentration of the microplastic in many of these locations will vary significantly because based on the land use. But LA, we didn't find that. Whether it's industry, proximity to wastewater, again, these are not a lot of data, when it comes to confounding factors. But at least it's an interesting result to see that entire LA is uniformly contaminated with microplastic anywhere you go. Then we went to, again, different sites, but this time it's all BMPs. Many of the green infrastructures are around there. These are the locations. Many of the locations we've been there, and we try to collect the microplastic on the soil and in the BMP sediments. Here is the results. When we examine as a function of depth, initially we know that most of the microplastics are retained in the top 5 centimeters. So we went there to see where they are. So again, it confirmed that most of the microplastics are retained the surface. We didn't say at the results, most of them, but this is just to show that many of them are retained in the surface. They can be resuspended in next time there is rainfall or flow events. We wanted to also understand if the impervious surface in the catchment area has a role in how much microplastic is retained in the system. Because our hypothesis is that if you have many impervious surface, they will cause also production of more microplastic because of abrasions, for instance, roads and all that. So we thought that maybe having more impervious surface, maybe more production of microplastic. That means more microplastic will find them in the stormwater BMP. We find very weak correlations. Again, the data is very scattered. We got more data, didn't analyze it all of them by the time we made this graph. But now the graph looks similar but more data. So what we learned that BMP, we are going to find a lot of microplastic no matter where you are, and we have to design such a way that it remove most of the microplastic. So I look at multiple contexts. First is what BMPs are made of. Most of the stormwater infrastructures, they replace the soil with sand and compost to increase infiltration. So this is one thing we did is we try to find out how compost types makes a difference in how much microplastic already present in the compost. So we collected many compost from locally available compost, commercial compost, and community garden compost, and we found that there are microplastic in them, which is expected because first many of the feedstocks may contain microplastics. But good news is that these microplastic did not ended up in the effluent water because most of the microplastics stay in the systems. So again, this is just the data that previously not known is the how whether the green waste, organic compost, or what kind of compost may have more microplastics. Finally, as I said, we try to see that whether these plastics will ended up in the effluents or not. So we did this column studies where we pack column with the sand and compost and some soil and see how texture of the BMP media influence the microplastic transport and removal. So this is just an example of showing that very small fraction of soil, now the 10% versus 40% soil. This is how microplastic ended up inside the columns once it deposits in the surface and have this stormwater pass through it. The 40% soil, which is the square one, the circle one, you can see that most of the microplastic on the surface, and then it become almost zero as you go deeper. So what it means that if you design this kind of BMP and add some soil or change the texture, it can have a greater influence in how plastics are retained in the system. So having some fine grained soil, I'm not fine, even small amount of soil would be helpful. Then what happened to the plastic which are retained on the surface? We expect that they will be resuspended. So we did some study. Again, I presented last time. This is just a column study just to show that we put plastic on the surface, and when we have high flow event, some of them are resuspended. What that means is any microplastic retains will not be retained forever. They will be resuspended at some point. Again, we got some comment last time that this is a column experiment where the flow condition is not similar to the real field. So we redesigned this study and simulate bioswale. But before that, I want to mention that in this study, we also learned that if you add some gravel on the top, which is a common practice, this gravel can flow down the water, at least at the interface, and retain more microplastic. So this is a good design conditions where any BMP, if you put any rocks or on the top of the soil or the filter media, you probably will keep the plastic over there. So again, this is a simulated bioswale experiment. You can see that this is just a plexiglass, and then this is a stormwater is being injected here, and we deposit some of the microplastic here. We observe how this moves through the system over time. And we created deliberately plastic which we can directly count and using microscope or just even visually. And you can see there are fibers and there are fragments so that we can see how shape influence it. So this is just again reconfirm the same thing, whether it doesn't matter what type of plastic. In the very first flush, when there is rainfall, that's the time most of the plastics are resuspended, or because of seep flow, they are being all removed from the system or goes to the effluents. And as the time elapses, you don't see much plastic in the water because most of them have already washed off. Here we wanted to show that these plastic which are retained in the bioswale, they could be retained in the edge or it could be center based on the plastic types or particular density. It turns out if the lighter plastic like polypropylene they are pushed by stormwater flow more easily, so they move to the side. So you might see every time there is rainfall, you might see a lot of these sediments and leaves and everything deposited on the edge versus center, and those edges may contain a lot more lighter plastics than the center place. So if you are going for sampling, center versus side makes a big difference. And this is also important for the maintenance, because you can always go to the side and remove them. This is just to show that fiber fragments or shape does matter. You can see that it has again, this more busy data, but what that means is fibers are retained consistently, more likely to be retained than the fragments, and again, it depends on where it is. So we had another studies where we had the field studies, the projects on different topics. So we went there and collected samples, and we also observed even a small design like this can remove a lot of microplastic, like having a retainers like this. You can see so much rubber tire wear particles are there. So again, what we learned? We learned that land use could be a factor, but big city like Los Angeles, you'll expect to find microplastics everywhere. So land use may not be a big factor because of the wind and everything redistributing them. And then we learned that if you can design BMP such a way that it can remove plastics, something like adding gravel or having having systems where you can change the texture of media filters. At the end, plastic will be resuspended. They will be relocated to the side or edge. That could also help maintenance in removing those locations for any plastic, so that it continue to remove more microplastics and not cause any pollution later. So there are many students who support this one, and these are just some of them. With that, thank you so much. Again, I'm very grateful for this opportunity.
Thank you, Sanjay. We don't see any specific questions for you in the chat just yet, but if you have any questions for Sanjay, please ask them. Oh, we did have someone just add in, just asking if your results are also preliminary or if these are final.
These are final results, but some of the data may change because we didn't put every data there. So the conclusion is not going to change. We have seen that.
Okay, great. All right. Well, thank you. And if any more come into the Q&A feature, would you mind just responding to them via written responses? Thank you so much,
Yes.
Sanjay. All right, so then our last but not least, we have Elizabeth Fossman Beck from the Southern California Coastal Water Research Project, representing her team and sharing the results from their project titled Field Monitoring of Microplastics Loading and Accumulation in Low Impact Development Best Management Practices. All right, take it away, Elizabeth.
Thanks very much, Tanya. Thanks everyone for being here. Tanya, I presume you can see my slides and hear everything okay? Great, thank you. So our title is a mouthful, so I'm not going to repeat it, but I am going to acknowledge there's a fair bit of jargon in there. So I'm going to take a second to try to orient everybody to what we're talking about with low impact development, best management practices, and urban runoff. Engineers often refer to BMPs as a toolbox of solutions for urban runoff quality and for urban runoff water quality treatment and flow management. BMPs are built to comply with provisions of the Clean Water Act, and those are to address regulated pollutants. So there's this whole slew of BMPs out there. They all work differently to treat pollutants, and they have different attributes and design features. That means an engineer, in theory, should be able to pick the right one for the job for a given site or a given location or given construction conditions. And you may not always choose the same type of BMP, right? So the BMPs, so that's BMPs in general. And the BMPs in our title and in track 2 were about low impact development BMPs This refers to a class of BMPs that are typically have a smaller footprint, and you'll find them more distributed through the landscape, serving the runoff from smaller drainage areas, like a few acres. And in my slide here, that tends to refer to the types of BMPs that are in like the top kind of pictures. So bioretention, which includes things like, you might hear them called biofilters or bioswales or planters, things like green roofs, tree pits, and actually even permeable pavement. That's in the bottom left. So that's what we're talking about with LID and BMPs overall. The contrast to an LID BMP, excuse me, is down here in the bottom right, where you have these really large facilities that might be underneath the ground that manage the runoff from like hundreds to, in some cases, thousands of acres of drainage area. But in this case, they're very out of sight. But anyway, we're focusing in on LID type BMPs, and very specifically, we're talking about in our study a particular BMP called the biofilter, which is amongst this larger group of what are called bioretention BMPs. So if you're not familiar with the bioretention BMP or how it works, let me show you for a second. So in what you see on the screen here on the left is a schematic if you're looking at the side or a cross section of a biofilter. And on the right, what it looks like in real life and in Southern California, which is not always super pretty. We like to think of BMPs as being these really beautiful features, but first and foremost, they're treatment systems, right? We're designing them for water quality treatment, and that's the most important thing from my perspective as a civil engineer designing BMPs to comply with the Clean Water Act and try to reach water quality objectives. Okay, our BMP in this picture is by the side of the road. That's the drainage area. So you can see it in dry weather and wet weather. And in wet weather, when it rains, this runoff comes off the road and it flows into our BMP. So that's our untreated runoff. And what it's supposed to do is that runoff builds up a layer on the surface. It does what we call as pond, forms a little standing layer of water on the surface. And the only way out for that runoff is to percolate through this engineered media. So it can only go down through this media. And when it's in that media, or as it's percolating through that media, two things hopefully happen. One is pollutants are treated by the media. And the second is that that media can also act like a sponge and just reduce the total volume of discharge that goes downstream. Notice that the word engineered is in there, and that means that we're actually specifying the components of what make up that filter media, and therefore we can manipulate the recipe and the design of that media to treat different types of contaminants. Of course, in this research program, we're looking at microplastics as our priority contaminant. So there were lots of things that we tackled in this research project, but I picked out 3 priority questions to talk to you about today, just in the interest of time. Number 1, how well do our existing biofilters reduce the amount of microplastics in urban runoff? What characteristics of engineered media promote microplastics capture in biofilters is number 2. And number 3 is, are there recommendations for future biofilter design or maintenance to mitigate microplastics pollution from urban runoff? And very quickly, let's start with the answers. What did we find? Well, first off, what we're finding is that in the biofilters that we monitored in Southern California, our biofilters are really efficient at reducing the concentrations of microplastics in urban runoff. Typically, we're seeing about 72% reduction in the concentration of microplastics between what comes in and what goes out. From a size perspective, particles sizes down to 20 microns were removed very efficiently. 20 microns was the smallest size fraction that we measured. And really in the effluent, the treated discharge, only the smaller sizes were detected. So all the larger stuff was removed very, very well. The characteristics of the engineered media, right? We can manipulate this by design. Microplastics are removed by physical filtration. And when we have media that had smaller pore sizes, in other words, very small gaps within the filter, media with smaller pore sizes accumulated more microplastics over the service life of the BMPs. The oldest BMPs we monitored were 6 years old. And And recommendations for future design or maintenance. Our biofilters, this was a super happy story and a great outcome. As a BMP researcher, this is one of the most positive stories I've ever had in my nearly 30 years of studying BMPs. Our biofilters were consistently and efficiently removing microplastics from runoff. Microplastics are only a proportion of the overall particles that were removed. And we didn't see any clogging or maintenance issues in the biofilters that were studied. And, notably, while I think they were removed really efficiently, there is the potential to manipulate the design further to further enhance removal. So that's the answer in case I run out of time. But maybe you want to know how we came to these conclusions. So let me walk you through some of those details. We leveraged an ongoing regional BMP monitoring program run by SQUIRP, that's the agency where I work, on behalf of a collaboration called the Southern California Stormwater Monitoring Coalition. We monitored runoff from 7 biofilters. The biofilters received runoff from roadways and highways to parking lots to parks, so lots of different urban land uses. And I said there are 7 biofilters that we monitored, but there aren't 7 photos because this one that's in the middle up at the top here, there were actually 3 of those along the same roadway, so replicates of each other. The samples that we collected were both from the runoff and of the media within the biofilter itself. The samples of the runoff, we sampled a total of 18 storm events across 7 biofilters. So it doesn't rain very often in Southern California, so we sample a few times at multiple locations to create our sample sets. Our samples represented really diverse rainfall characteristics, so we feel like we have really representative samples. And we collected inflow and untreated runoff and treated runoff, so influent or inflow and effluent or outflow samples from these biofilters in what's called flow-weighted composite samples. This is the gold standard for BMP monitoring, and the analysis of these samples give us what are called event mean concentrations. Lots more jargon for you, but rest assured, these are the gold standard. We also went out and collected media samples once from each of 4 of the BMPs at multiple locations. Sample processing has been pretty well touched on by everyone else, so I'll skip right over that to get to the details of the numbers. The graph that you're looking at right here gives a representation of our treatment efficiency. On the x-axis or the horizontal axis, we have the untreated runoff concentrations. And on the y-axis or the vertical, we have the treated runoff concentrations. That dashed red line gives you the line of equal value. Data points to the bottom right corner, to the bottom right of that, mean that the influent concentrations were greater than the effluent, meaning that there was treatment happening. You can see there's only 1 dot on the other side of that line, meaning there was more microplastics going out than coming in. We're not going to spend too much time on that, but suffice to say, seeing that kind of result is really typical for BMP monitoring. BMPs are highly variable most of the time for treating most pollutants, but what we found with the microplastics was that our typical removal efficiency was really consistent, ranging from, for most of the data, between about 54 and 89% per storm event, and overall a typical treatment of 72% concentration. From the size distributions, these were super fun slides to look at. We see every size fraction from 20. Again, our smallest was 20 microns. Our largest was greater than 500 microns. We see every size fraction represented in the inlet. I think Matt also talked about how the smallest size fractions are the most abundant. That held true here as well. In the effluent, we really only saw the small size fractions in the effluent. But even though we saw them, they were still significantly less than they were in the influent. Now, I mentioned that there's engineered media, and that means we can manipulate the design of that material itself to make a better filter or make a filter do what we want it to do. And for particle capture, what we want is really small gaps between the particles that make up the filter itself. The smaller the gap or the pore size, the smaller the particle that can be trapped. So in our study, we went out and collected media samples. As Sanjay identified, you need to collect from multiple places within your BMP. So we collected from multiple places and mixed them together for a representative sample. When we set up this study, we said, right, we want to sample the media and measure the sizes of the gaps or the pore sizes within that media, because just in case the BMPs don't capture microplastics, we want to know if it's because of the design of the media. Now, fortunately, our BMPs were really great at capturing microplastics overall, but we absolutely saw that when we had more pore sizes that were smaller than our smallest particle we were measuring. So when we had more pore sizes that were smaller than 20 microns gap size, those BMPs did a better job of removing smaller particles. Just confirmed what should have been obvious, but it was really worth the deep dive inside the BMPs. Okay. Bottom line is, to remove the microplastics, our pore sizes have to be really small. Okay, the implications of our study and next steps as we see it are that the design of implementation and biofilters, the existing infrastructure that we're already building, seems to yield consistent and really efficient reductions in microplastics that might otherwise be transported in urban runoff to receiving environments. This just gives another tick in the box of why should you choose a biofilter from the toolbox of BMPs where you have multiple BMPs being considered. Microplastics, right, are an unregulated contaminant, so they're not currently in the mindset of engineers to choose from or site owners to choose from, but this could be another bonus feature. We also think that other BMPs that use physical filtration to remove pollutants are also likely to be successful in capturing microplastics. So, because of the consistency of the results from across these 7 biofilters, we feel like we had a really good representation of biofilter performance, and it's worth having a look at other types of filtration-based BMPs. And my #1 choice would be permeable pavement, right? Permeable pavement acts right at the source, and especially where tire wear particles might be a concern, that would be a good one to look at next. Ultimately, while we have great capture efficiency, smaller microplastics do discharge from biofilters. We did not look at receiving water impacts. And of course, as been mentioned by several of the researchers before me, the toxicological effects tend to be thought to be from the smaller particles, and this really should be looked at. So we have efficient capture at the site scale, but is it having the beneficial impact on the receiving water that we need? We didn't measure that. But if you need to capture more small particles, there is an opportunity to fine-tune that engineered media design. With that, I'll say thank you. If there is a chance for questions, happy to answer them. On behalf of me and my colleague Danwei Shin here at SQUIRP, the primary researchers involved, as part of this research program, we worked with Cal State Long Beach and Cal State LA to both host workshops for the students, but also to recruit student employees. We had everybody showing you how the analysis is huge in the laboratory. We had this fantastic army of student researchers who are still working here at SQUIRP, even though our study is complete. Really great opportunity, fantastic students, and we have really appreciated their devotion, effort, and just attention to detail in doing this. And of course, a big thank you to Ocean Protection Council, Sea Grant, and the SMC for supporting the study. Thanks. I think I might have gone over. Apologies.
No, you're great. Thank you, Elizabeth. That was great. We do have a few questions. I know we're moving into our panel soon, so just we'll get some of the specific ones for you out of the way. There was a question that came in, interested on how biofilters are cleared once they're saturated.
Okay, so I'm going to interpret that question a bit. So I think the question is really about the long-term maintenance. So over the long term, yeah, you have a filter and it's accumulating pollutants inside. That's what it's supposed to do. It's doing its job. You can do kind of routine maintenance. So on a 5 to 10 year basis, it depends on the loading. You might need to go in and scrape out the top layer of your biofilter to restore the capacity. But over the lifespan of a biofilter, which no one firmly knows yet, but it might be on the order of 20 or more years, there may come a time for restorative maintenance where you actually have to go in and scoop out the material and replace the entire thing.
Excellent, thank you. Let's see. Okay, and one more we have specifically for you. If we drop down media size, doesn't that decrease the amount of water filtered, meaning more water might go through overflow valves during heavy rain?
Yeah, that absolutely could be the outcome. So there's you walk this fine balance of adequate filtration rate and infiltration rate versus capture. So that's not so the suggestion that we can fine tune or the idea that we can fine tune the media for to capture smaller particles does come with the warning of you need to make sure that you're evaluating the BMP as a whole and whether or not you will still capture enough water. So it's not a just go out and do it. It does need to be studied.
Gotcha. Thank you. And there are a few more specific questions for you, if you wouldn't mind. But right now, we are going to bring back our other panelists. We have about 10 minutes to open the floor. Um,
I'm
for
sorry, Tanya, I'm not seeing these questions. Am I not looking in the right place?
Oh, it's in the Q&A function. There should be at the
Oh,
bottom.
sorry, found it.
All
Thanks.
good, all good. But we're gonna ask our all of our five panelists to turn their camera back on, and we will spotlight you. We wanted to have a panel Q&A before we end our public portion of this webinar. Awesome. Waiting on just Sanjay. Perfect. Perfect. Thank you. Okay. And so before we open it up for questions from the audience, we did want to just start off with a priming question. And so, in your own words, briefly, we'd love, you all just gave great presentations, a lot of information. We still have about 100 people on this call. And so we want to hear from you why do these research findings matter to Californians? Like if you could share more about your specific research and why all these people in California should care about the work that you're doing. And so maybe we can start with Matt. How about you? Sorry to put you on the spot.
Yeah. No. It's yeah. No, I was thinking about it as you asked the question, and I think the data that we're collecting and showed briefly talks about environments that we all use, environments that we live next to, that we visit, that we enjoy, and it can help teach us about how to be better stewards to that environment. Whether that's actually going out and participating in a cleanup or just being really mindful about what you bring, for example, to the beach and what you take off and how you sort of leave no trace, as they say in the Boy Scouts, which I was a part of when I was a kid. So, not to say that I'm perfect at it, because I'm certainly not, but of course I could do better. And we're all playing a role in this, and so I think thinking about these ecosystems that we use a lot, both for recreation, commercially, tourism, and so on, and how we impact those environments, even in ways we don't see, I think is an important revelation of some of the stuff that we've shown.
Thank you. Yeah, Diana, you're next.
Well, microplastics are really diverse. We're seeing them everywhere, and the sources of them are very diverse. And so I think it's important to identify what the major sources are so that we can focus our attention on uh, solutions that will make the biggest difference. And different sources of microplastics it's gonna require various strategies to address the different types.
Yeah, we see that definitely through your work. Thank you. Anyone else want to take a stab at answering that?
For my project, obviously there's a lot of Californians that eat fish. There are also a lot of divers and other people that rely on there to be healthy ecosystems. So our work really gets at how long plastics have been in these systems, potential ways that you might be exposed to microplastics if you're eating some of these fishes, and really getting that historical baseline so that you have a metric to go by as we move forward with possibly implementing some of these better mitigation strategies.
Thanks, Bill. Sanjay or Elizabeth, did you want to share?
I can comment in that, the question for us was, and this is part of the microplastic strategy, is what are we already doing that maybe we could do more of to that would address microplastics. And BMPs are installed to comply with existing regulation. So the driver for BMP implementation currently has nothing to do with microplastics. But we have this, what our study is showing is that we actually do get well, from a BMP person standpoint, we have a really great benefit. So perhaps there's something to build on there.
Yeah, thank you. Sanjay, anything to add to that?
Yeah. I just want to add that is one of the major states with the biggest coastline, and so we pollute our beaches by different pollutants. Microplastic is one of the big one. And what we don't really address in these 5 presentations that these microplastics are not alone. They carry a lot of pollutant. They are attached to a lot of other contaminants. That makes them very toxic to fish and ecosystem. So removing them is not just removing plastic, removing many of the other pollutants they may be carrying. So it's a great benefit in general for ecosystem, human health, and all that.
Thank you so much. We're going to move on to another question. I know some of you mentioned this throughout your presentations, but we're curious if you could share what is challenging about doing your type of research and what hurdles maybe are you experiencing? And I know maybe there's some really technical ones or maybe there's some broader ones. So we'd love if you could share a little bit on some of the challenges you've been experiencing. And
Analysis.
yeah,
That's it. I mean, that's it. I'm sorry, I totally just jumped in, but this is the biggest challenge. The time and the expertise and the time and the expertise and the associated cost of analysis is enormous. If we really want to get a good handle on microplastics in the environment and sources and pathways, fate and transport and solutions, if we really need to bring down the cost and the technique and improve the techniques that are used for more rapid assessments. I mean, you heard almost everyone say there's an extension to the study, and I think what I heard was that it was all because of analysis, laboratory analysis. Once we get the data, we can interpret it.
Yeah, definitely seconded, and that's definitely true for our project. Another way to frame that though is to increase funding for contaminant related research, right? And to put contaminant related research on the same pedestal that climate change or biodiversity loss has in terms of our how concerned we should be about it and the resources we should dedicate to dealing with it. Ideally, it would be great if we could get some of these costs down, but I think plastics, even though they might seem simple to lay people, like we know what a plastic item is. I mean, when they get really, really tiny, these instruments we're talking about, Raman, FTIR, stuff like this. These are instruments that are dealing with micron or submicron in certain cases scale particles. And so if you have instruments that are off in terms of their alignment by 1/100th of a human hair, they need to be fixed, right? And that takes experts to fix them, time to fix them. And so it's just I wonder about this, Elizabeth, and I talk about this like how reasonable is it to bring down costs in like a really significant way when it's just like really complicated pollutants to deal with, right? And I don't anyway. So either bringing costs down, bringing funding up, some combination, but it does seem to be this analysis thing for sure.
One of the things that happened during the course of our study was that actually Wynn Cowgar from the Moore Institute developed some mapping techniques that automated some of our FTIR steps. And that we actually tracked And that saved a huge amount of time in our analysis time. We actually tracked the time required for every step of the analysis, and it having the automated mapping cut the analysis time nearly in half. That is $1,000 savings right there per sample,
Mhm. Yeah,
or per
Wynn's,
size
Wynn's,
fraction per sample.
yeah, Wynn's great. We're lucky to work with him. We all are.
Yeah.
He's a pioneer in this stuff. So yeah, it's been great. He's awesome.
There are always trade-offs though. So there's trade-offs with it, how you analyze anything. One challenge that I don't think has been brought up, that I think maybe some people on the call, if they don't work with plastics, might not also have thought about is contamination. We spend a lot of time thinking about reducing contaminants in our filters and our data. Microplastics are everywhere in the world we live in, and it's really easy for microplastics to contaminate your data. So there's a lot of care and thought, I'm sure, on all of us on this call that we deal with to reduce contamination as much as we can. And that is definitely a challenge of doing this work.
Yeah, I was just going to second everything that Elizabeth, Matthew, and William said. And also, like, this was a pretty unique funding opportunity for our study. We had been uh, developing the study idea for a while, and it was really great to see OPC and Sea Grant be really interested in this topic and provide this funding opportunity.
I have one more piece to add to this analysis time thing. Uh, we also, so in our analysis, we checked when we looked at the metrics that came out of the microscopy. So just when you count all the particles, they correlated super strongly with microplastics. And so meaning that microplastics were a consistent fraction of the total particle count in the urban runoff samples for both treated and untreated runoff. And so for at least BMP samples, I would strongly recommend that in future studies, we can actually reduce some of our cost by going through the microscopy step for all samples, but only going as far as spectroscopy for some of the samples to just check those relationships. It held true for the total counts and also for every size fraction that we looked at. And so this would be a big savings in future analyses.
Great. Well, that kind of leads into my final question. We have 1 minute left, so this is going to be a rapid round. But based on what you've learned so far in this research, what would be your next course of action to further this, or just maybe some lessons learned? And as brief as you can, and maybe we'll start with Sanjay.
Yeah, I think the next course. I mean, having funding is the best thing because we don't really have enough of research. Plastic is something is very new areas, and California is like doing a lot more than many other states. So I think this is the first thing: having more funding and asking these questions. Group of researchers sharing results because the analysis is the biggest part of the challenge. But everywhere we learn, we can share the results so that other people don't go through the same process. So I think more collaborative work is much more important than just doing it randomly in a different place. So I'm grateful that all five of us or other people joined together. What Elizabeth shows that what I've seen in the lab results. So these are very important to learn from each other.
I'd say
Great.
for us, it's sourcing and risk, getting more specific with where our microplastics are coming from, the ones that we find in the environment, and the specific risks they pose, whether they be physical, chemical, or otherwise, to ecosystems and organisms. We've applied for some funding to do that work. Hopefully, we get it, but sourcing and risk and getting more precise with that.
I'm gonna echo that. I'll jump in. Toxicology, the toxicological impacts of what's actually getting into the receiving environments, developing more thresholds and looking at, we may have some solutions, but are they making an impact?
I guess I'm interested in following up on this was kind of our first pass at our study question, so refining that and looking more into the air transport pathway and trying to understand like the importance of local sources like drier emissions in our urban landscape or global transport. Kind of what's the bigger impact on local air burden of microplastics?
For me, I think one thing that I'd like to follow up on has already been brought up is we're looking at the guts of these fishes. What are the plastic loads in other tissue types? I think is really important to look at in an obvious follow-up. I'm also really interested. I mean, this this study has raised so many open questions too. I really want to look at larval fishes too. We have large larval fish samples as well, because those earlier life history stages are also very vulnerable, very high mortality rates. And if there's not enough fish to settle because they're eating microplastics, you could have very large impacts on population numbers. So that's another avenue that we'd like to look at. But there's so many open questions, especially the toxicology question is really important to look at. So I'm going to echo that one as well.
Well, thank you all so much. This does conclude our public portion of this webinar. I do know that there's a couple questions left in the Q&A if our researchers wouldn't mind just double checking that you've answered anything that you can. We will be kicking out almost all of you participants, but thank you so much for joining, and our researchers hang on because we will be moving into our closed door session in just a moment. But thank you so much, and you can reach out with any questions. Maybe Kyla, can you drop our emails in the chat?
Yes, I can.
Yeah. Oh, and let you know there is a feedback survey that will be shared through Zoom if you could